Hello, my name is Michael, and I'm a bioinformatics PhD student at UCLA in Dr. Xia Yang's lab. I'm presenting my project called scGRNdb, a cell-type gene regulatory network atlas for human and mouse. Gene regulatory networks try to model the mechanisms in network mechanisms in different cell types or tissues, and it's proving to be very important in understanding cell heterogeneity with the advent of single-cell RNA sequencing and other single-cell data platforms. We know that many single-cell data repositories are only rapidly expanding, increasing the number of data sets that it hosts, and this highlights a critical need for robust GRN methods to analyze single-cell data and also a comprehensive resource to host cell-type reference GRNs for other researchers to use. So we developed a single-cell integrative gene regulatory network method called scGRN in our lab to build these GRNs from single-cell data, and we're applying it to a novel database called scGRNdb, which hosts cell-type GRNs and network analytical tools, leveraging millions of cells across these single-cell data atlases. First, I'll talk about the method scGRN and how it works. So scGRN uses, again, single-cell RNA sequencing to build gene regulatory networks for within each cell type. So with pre-annotated single-cell RNA-seq data, we would perform a lead-in subclustering on each cell type to generate 500 subclusters, average the expression within each subcluster to get pseudobulk cells, which mitigates the gene sparsity known to be a technical effect in single-cell RNA sequencing. And we would perform a bootstrapping of 100 random subsamples within these pseudobulk cells and build a network on each bootstrapped sample. The network inference is performed using gradient boosting, which is a tree-based machine learning algorithm, which trains shallow decision trees in an iterative manner, where each decision tree is trained to predict the errors of the previous ensemble of decision trees. That way, as you continue to add decision trees to your final regression model, you're more closely approximating the target variable. So we train a gradient boosting regressor on every gene in our data set. We limit the feature genes used in each regressor using a k-nearest neighbor approach in the cell principal component space, and this reduces the model complexity when building these networks. Now, performing this on every bootstrap sample gives us 100 different intermediate networks, each having spurious edges. We want to look for highly consistent edges that are conserved across all of these subsampled networks. That's the point of this bagging approach and these different filtering steps. It's just to reduce false positive edges and strengthen the biological signal in the networks. So with this algorithm, we're applying it to many cell types and single-cell RNA sequencing data sets from many human and mouse atlas repositories. Here's a list of the atlases that we have been using, and we are continuing to add databases as we go. So far, we have 1,019 unique cell-type GRNs across 90 tissues and 2 species. These networks have an average size of about 15,000 nodes and 50,000 edges. Along with hosting these networks in scGRNdb, we also plan on hosting network analytical tools to interpret these networks biologically. One of these methods would be an enrichment analysis. From a given cell type GRN, we would perform a leading clustering on this graph to get highly connected subnetworks, which we call modules. These modules we will use in a hypergeometric test to find enrichment with biological gene sets, such as disease-related genes or pathway-related genes, and annotate these modules with these significant enrichments. Here we show an example of a mouse primary motor cortex excitatory neuron network, where we run this enrichment analysis. We see that these modules contain very distinct biological function that is relevant to neuronal function, such as module 7, involved in neuron and synapse development, module 9, calcineurin-mediated signaling, and 14, which is involved in regulation of vesicle-mediated transport in the synapse. Now, the enrichment analysis really scans the network topology and finds biological meaning. We also want to provide a network tool that guides a disease study. For this, we propose key driver analysis, which is another form of enrichment test, which finds an enrichment of disease genes within highly connected subnetworks in your GRN. The idea is to propose key mechanisms that are shared between your disease genes, and the key drivers within these network hubs, or these very central nodes connecting these disease genes, will be the ones proposed as these key drivers regulating this disease. So we performed this key driver analysis on cardiovascular disease gene set known from this DisGeneNet database. We performed it on a liver hepatocyte network. Running this key driver analysis returns some key drivers that are related to cardiovascular disease, as we see in the blue color, but also some novel genes that have been implicated in other diseases like non-alcoholic fatty liver disease in the case of AGXT. These genes are also very specific to the hepatocyte function, which is a good validation for us to see. When querying these same disease genes on a macrophage liver subnetwork, we see very genes related to inflammatory signaling and inflammation, which again gives a good indication to us that these networks are capturing true biology. So in summary, we've introduced scGRN and scGRNdb as important tools for network modeling and understanding cell type contribution to disease. As a future direction, we hope to perform a systematic benchmarking and evaluation of our networks, looking at GWAS summary statistics and genetic mechanisms across different cell types and tissues. We want to include co-expression networks and other types of networks in our database, using methods like HDWGCNA, and then also build a web server to host these networks and analytical tools, and to make visualizations that are very user-friendly and that really cater to the needs of bioinformatics research. But thank you for listening, and I'd like to acknowledge our funding as well as other members in the lab that have been very helpful for this project. Thank you.